Good morning to everybody. I'm Roberto De Carolis and my research topic is focused on lichen photobiont diversity in extreme environment. The last 10-year studies about photobiont diversity allowed us to understand the importance of photobiont in lichen symbiosis. From this study, we have learned about the important role of photobiont diversity play in lichen distribution. Also, the importance of specificity of photobiont toward microbiome species in the lichen ecology, and a deep knowledge of photobiont diversity can help us to understand the tricky relationship within symbiotic partner in lichens. To uncover the tricky photobiont diversity, in our project we use an integrative taxonomic approach, and we will reach two main aims with our project that are uncover the photobiont diversity entirely from extreme environments, mainly in oligotrophic and high altitude conditions, and correlate the photobiont diversity with the specificity toward the associated microbiome and ecological condition. To do that, we have chosen two cosmopolitan lichen species as model, Rhizoplaca, melanophthalma, and Tephromela, atra, and collected the sample in both south and north hemisphere from extreme environment. We did algal isolation in axenic culture from thalli and performed the molecular analysis of ATS locus for phylogenetic reconstruction. The axenic culture are also used for classical morphological analysis in light microscopy and for ultrastructure analysis in transmission electron microscopy. We also obtain ATS locus sequences from thalli directly to compare them with ITS sequences from axenic culture to find the co-presence of Trebouxia species in the same thallus. So far, we have already analyzed 190 ITS sequences that are discovered in two of the four main Trebouxia clades, clade A and clade I, and we observed that in 23 of these 89 thalli analyzed, two or more Trebouxia species co-occur. A first ecological analysis shows us that Rhizoplaca so far seems to be the more generalist lichen than Tephromela atra, but to assume that we need to collect more data. And the Argentina is the area in which the greatest diversity of photobiont is recovered so far. Focusing attention on the clade A, in which we recovered the majority of our new sequences, you can see on the left the phylogenetic reconstruction, where I marked in green the lineages that include our samples. Is to be noticed that we have a large number of our sequences that classed in lineages that are not described yet. The most important of them is the A02 clade that include 53 our new sequences that came from just from Rhizoplaca melanophthalma thalli, and these thalli are collected all over the world. Then we have just one sequences that class in another not already described lineages, the lineages A16, and the other sequences are included in already well described species. But the most important things that I want to show you is the presence of 16 our sequences coming from Argentina that class in new lineages that is reported for the first time in this study, and can be potentially a new species. So on the right you can see light microscopy image of these three lineages where is visible the morphology, the differentially morphology of chloroplast of these lineages. On this stage, we are still gathering new samples for implementing our dataset. At the same time, we are performing the transmission electron microscopy for potentially new species and for samples belonging to not yet described OTUs. The final step of our project will be the ecological studies to estimate photobiont diversity pattern, study community structure in extreme environments, and find correlation with specificity for the microbiome and ecological condition. Thanks for your attention, and a special thanks to my PhD supervisor Lucia Murgia, and if you have any question, I'm here for you.